All right, thank you all for your attention today. You know, I think in addition to that caption contest, we need something to figure out what's going on with the dog, the politician, and the toxicologist. That's really, I gotta know. So it's my pleasure today to talk to you about one of the projects we have ongoing within the Genomics Committee. This is a project focused on understanding microRNAs, and it's really kind of working towards biomarker discovery. I do have to point out that Aaron Smith, who is one of the leaders of the subcommittees for this project, actually helped with putting some of these slides together. Unfortunately, couldn't be here today, but was an integral part in the messages that I'm going to deliver to you. So why do we care about microRNAs? So microRNAs have generated a lot of enthusiasm in recent years for many reasons. One is that it's a much smaller biomarker space as compared to mRNA, so we're talking about hundreds or a few thousand versus tens of thousands. They can be sensitive and specific biomarkers, as have been shown in many studies. They can be easily measured in biofluids, and they're relatively stable in serum and plasma. So there's a lot of advantages over protein biomarkers as far as stability. They're highly conserved across species depending on the specific microRNA. So you can really develop just one RT-PCR assay and one set of primers, and then you're good to go for both preclinical or non-clinical and clinical species. And in addition, there are variants of microRNAs, which are called isomers, and they may offer additional space to query for biomarker candidates. MicroRNAs, I should mention, are just very small, short sequences of microRNAs that have a function for post-transcriptional gene regulation. So in practice, when we're thinking about using microRNAs for toxicology, which is where we're really sort of focused in the Genomics Committee, we think about a variety of different ways, and hopefully you can see my arrow here, maybe not. There's a variety of ways that these microRNAs can be released into plasma, urine, or other biofluids. You can have active secretion through a variety of mechanisms. You can also have passive secretion through disruption of the cellular membrane, as would occur during cellular injury or toxicity to a given cell type. So those are some reasons why we're interested in microRNAs. In the context of our committee, we see a lot of opportunities for using microRNAs as biomarkers. So one of those is being able to fill gaps where we want to be able to get information about an underlying tissue, but there are no currently good biomarkers that we could use other than looking at histopathology, but in vivo we can't have a good read on what's going on within the tissue in a specific context. We want to decrease the time and cost of development, particularly of drugs, but also chemical products. We want to enhance clinical translation of biomarkers, and microRNAs, as I mentioned, are very useful in this context. And we want to improve sensitivity and specificity. So can we detect organ damage very early before some of our gold standard protein biomarkers become elevated? So we're really, in this context, sort of applying the HESI model to frame what we want to be able to study here. And so this slide really lists a lot of the expertise that we have in our committee, and we're really drawing on all of these areas towards microRNA biomarker development. So we've got all the right expertise to address the issue. In addition, we have a strong history within the Genomics Committee of successful collaborations. We've had several subcommittees in various contexts. We have expertise on platforms for measuring microRNAs, so we thought this was a good area to target. And as far as microRNA biomarker discovery, in the context of this particular project, we can use new techniques such as next-gen sequencing to identify microRNA that are unique to or enriched in specific tissues, and here we're talking about the rat. And our intention is that having an atlas of these microRNAs and an awareness of the basal expression of microRNAs within given tissues can actually help lead to biomarker discovery and confidence that a given microRNA biomarker in a biofluid is derived from the tissue of interest. The other nice thing about this particular project is we see a lot of opportunities for cross-connection across committees, because here we're really trying to identify microRNAs that are specific to tissues. Other committees are working in other tissues as well. So there may be opportunities for collaboration here. So in this particular study, we actually queried 23 distinct tissue types. As I mentioned, we performed next-gen sequencing, or I didn't, the subcommittee did. And here you can see a list of the tissues that were collected from both male and female Sprague-Dawley rats. The next step was actually starting to figure out, well, okay, now we've got all this information about microRNA abundance in these tissues, but which ones are truly tissue-enriched or tissue-specific? And there were 3 universities involved in the data analysis, 3 institutions, so Maastricht University, Eli Lilly, and the NIEHS. And I'm not going to go through the data here, but these are showing you the specific microRNAs for each tissue and how many were identified by each organization. And what you can see here is that the data analysis pipeline actually had a very major effect on whether a microRNA was called tissue-enriched or tissue-specific. And so some of these data analysis and bioinformatics elements were very important to our understanding of the microRNA expression in various tissues. So these are actually some data that were provided to us from Eli Lilly, from Aaron Smith. And you probably can't read this too well from the back, but essentially what you're looking at is in red are the number of tissue-enriched microRNAs within a given tissue, and in green are the number of tissue-specific microRNAs, so they were unique to that particular tissue. And so you can see, depending on the tissue type, there's a varying number of tissue-enriched and tissue-specific microRNAs, and all of these data have been put together into a publication which is either has been submitted or is soon to be submitted for public dissemination. Okay, so the other aspect of this I wanted to point out is that we can make these data available to other committees. So again, we're very interested in cross-committee collaboration on this particular front, as well as others. Okay, so what lies ahead? So we've completed our study looking at the microRNA body atlas. We've started to scope out kind of a second phase of work, which we're kind of thinking of as a microRNA atlas phase 2. And so we've kicked the tires on various ideas. In our first round, we talked about and that was what I just presented to you, was characterizing the tissue expression of specific microRNAs to help guide some biomarker discovery efforts and qualification. In the second phase, we've thought about various ideas, and I'm happy to chat with other folks if you have other ideas or other areas of investigation that might be particularly interesting. But we're looking at, so we've done whole organ assessment. Should we get more detail on specific cell types within an organ, get a little bit more granularity on information content of a particular tissue? Should we explore the kinetics of microRNA release into various biofluids? For example, I'll show you some data that will indicate to you that timing of release, clearance rates might actually be particularly important in measurement and analysis. So those are some ideas. These are just some data that help to sort of give you some information about why we're thinking about these particular areas. These are some data that were generated by Aaron Smith at Eli Lilly, and he was actually looking at tissue expression or cell type specific expression of microRNA 122, which is highly enriched in the liver. And what he found was that, and you don't need to pay too much attention to this slide, I'll just hit the highlights, but he found that 122 was more highly enriched in hepatocytes versus bile duct epithelial cells. So seeing a release of 122 into the plasma or the serum might give you more information about hepatocytes versus the biliary cells, for example. And then as far as kinetics and timing of release, here's some data that my lab published very recently looking at microRNA profiles in plasma following a single administration of ANIT, which causes cholestatic liver injury. So we gave the ANIT at time 0, and then we actually tracked protein biomarkers and histopathology over time, with the peak of injury being about 48 hours post-dosing. And here you can see that at the highest, the peak injury time, you see a lot of microRNAs that are released into the serum, one of the most abundant being miR-122, which is sort of the poster child for microRNA biomarker discovery because it's highly liver specific, it's very stable. But actually what we see is if we look sort of earlier in the injury time course, as well as when injury is still apparent but resolving, 122 isn't the most abundant microRNA in plasma. There are other microRNAs that are actually more informative at those particular time points. So measurement might be measurement timing might be an important area of focus for our committee moving forward. So we're having some discussions now around where we'd like to go with this. Okay, so just to sort of kind of bring this all home, here's what we see as the impact of the microRNA body atlas as a whole as far as the work we've done so far. So the key idea is we want to develop this concept called a liquid biopsy where we can measure microRNAs as well as other factors in the plasma and have some information about the underlying pathology. Again, we'd like to translate these biomarkers across species, so are there implications for clinical use? We think so. And in particular, we'd also like to understand whether some of the microRNAs we're seeing as tissue-enriched could be informative for disease states. For example, here I'm listing on this slide just several microRNAs that have been measured in biofluids that are known to be involved with, certain cancer types, certain toxicities, acute myocardial infarction, for example, kidney injury, et cetera. So I think there's a lot of implications here that the microRNA body atlas really forms a foundation for. Okay, so with that, I know I don't have a lot of time, so I'd just like to acknowledge our working group on this topic, particularly Aaron Smith, Carol Thompson, and Hydron Ellinger, and then all of the folks across multiple institutions who have been involved with the RNA-seq data analysis and generation. So thank you for your attention. Yes.
Thanks, Al. That's really nice. But the data analysis, that kind of caught my eye on the slide. So given you had 3 different people doing 3 different approaches to their data analysis and coming up with largely 3 different sets of results that kind of overlap a bit in the middle, how did you decide which was the right result?
Yeah, that's a great question. I wasn't involved with that piece of the project, so I couldn't speak to that point specifically. But it's definitely something that we need to explore in a little bit more detail because it's very concerning. If you've got some lab that's running one type of analysis, another lab that's coming up with a different answer, how do we converge in the middle? And I think, and other folks can correct me, is that we, for this particular project, focused on where the intersection was, but we might be missing important information if we exclude some of the non-overlapping microRNAs. So it's a reasonable point, and I think one that we really need to look into in greater detail.
I think it's particularly important given all of the press today about reproducibility of results. If somebody's doing an analysis and someone else can't reproduce it, it's worth they're doing their statistical analyses, and is that playing into it?
Yeah, I think that's right. I think Jim in the back has some clarification on Lily's data.
You need to separate lack of reproducibility from trying to call very short sequences based on what might be a microRNA or might not be a microRNA. So these aren't lack of reproducibility. This is just different degrees of stringency and tuning of the algorithms to try to call these short sequences accurately.
Question in the back?
Yes. A couple of commentary or potential areas. Cell-specific nature of mRNAs. Is there an opportunity there to be able to get down to a cellular level with these? Is the first point. And the other point is about the
liquid biopsy piece. And here you're looking at it from a toxicology perspective, but what about the diagnostics field? Is it more accurate diagnostics and disease subsections of diseases?
Yeah, so as far as your first question, getting at cell type specific microRNAs, the HESI Genomics Committee has actually been focused on this area for the last several years, looking at whether or not we can identify microRNAs leaked into the urine that are very specific to specific areas of the renal nephron. And there are some very good biomarker candidates that have come out from that committee. Ernie, don't allow me to misspeak on the committee's work, but there's also a large effort into a meta-analysis to look across studies that have been converging on these different areas to see where the overlap is. So I think there are a lot of efforts in that space. Whether or not it's going to be organ specific, I don't know. There hasn't been enough data collected yet, but I think there's some promising early data that suggests that we might get there, at least for some context. For your second question, the concept of liquid biopsies and using them clinically, I think the issue is going to be simpler in some respects for toxicology, where we're starting with, for example, healthy animals, and then we're seeing microRNAs leaking out. Clinically, there may be issues with the underlying disease. So there could be microRNAs that are, kicked out into the plasma, that are related to the disease state that aren't specific to, a drug response that you're trying to look at. So I think there's possibilities there, but the field is so new, we haven't quite gotten to that point yet.
On the same theme, though, can I ask you, Alison, there are two things that struck me about the data and its potential utility was the brain and also miR-122 in the liver. But with the brain, I was impressed when I first heard that by quite a large margin there are more enriched and specific microRNAs in the brain than any other tissue, and you've done some regional distribution. Do you think that that's a rich vein to be pursued, to look at more specific distribution of microRNAs in particular areas of the brain?
Yeah, definitely. And I think there are folks who are working on it in other contexts, so not specific to toxicology. But I think that, some of the data that we have may help to guide very specific targeted studies to that point.